Hello, it's a late fall weekend morning on the Caltech campus in Pasadena. We're looking toward the northeast at scenes that may be familiar to viewers of Numbers. Now we're looking toward the north at the Kirchhoff building, these arcades and cloisters figured in several Numbers scenes. We're moving toward the west now, looking at the Bechtel Mall. And then we'll move toward the south and begin telling you about the Synapse as a drug-sensitive biophysical machine. Just reminding you that in the presynaptic terminal, we have vesicles full of transmitter, some of them docked to the membrane, awaiting calcium flux, which allows them to fuse with the presynaptic membrane and liberate their contents of transmitter into the synaptic cleft. Here in this electron micrograph, we see a presynaptic terminal and a postsynaptic cell, and we imagine that all of the action is taking place here in this 500 angstrom gap between the presynaptic and postsynaptic cells. In favorable cases, we can actually record simultaneously from the presynaptic and postsynaptic neurons, for instance, in the squid giant synapse. And so here is a presynaptic and postsynaptic recording, rather stylized. We have the usual action potential, and then the after hyperpolarization, and then the postsynaptic recording. If we did not have a postsynaptic spike, we would have the postsynaptic excitatory postsynaptic potential, which would reach threshold for a postsynaptic impulse and fire a spike. Back in the presynaptic terminals, we have the inward calcium current, which is very much responsible for transmitter release. Now, the whole synaptic delay between the peak of the action potential and the start of transmitter release is about half a millisecond. And actually the delay between the peak of the calcium current and the beginning of the EPSP is only about 0.2 milliseconds, a bit more at lower temperature. And so most of the synaptic delay is caused in the opening of calcium channels during the presynaptic action potential. And the size and timing of the EPSPs can be modulated by changing the waveform of the presynaptic action potential, and several drugs change transmitter release in this way. For instance, ziconotide blocks these calcium channels. Ziconotide is a relatively new painkiller. It's one of the few peptide drugs that work in the brain, and it needs to be introduced in the brain with an intrathecal pump that is inside the spinal cord. Moving on to ligand-gated channels, we're going to talk about the timing of their activation today, reminding you that they are multimeric integral membrane proteins, that ion flux through these channels can send a signal in two ways: a change in the membrane voltage or calcium entry. This is critical for transmitter release, for synaptic plasticity, and for several other roles. Indeed, calcium is a very common universal transmitter in the nervous system. Cells typically maintain their calcium concentrations quite low, and this allows them to use transient, localized calcium entry as a signaling mechanism. The channel opens within just a few microseconds after binding a ligand. There are two types of ion selectivity in ligand-gated ion channels. There is cationic selectivity, and as we'll see, this is often non-selective for sodium, for potassium, and sometimes for calcium as well. And then there are anionic postsynaptic receptors, which flux chloride at GABA-A and glycine receptors. And these proteins are subject to modulation in various ways. I'll remind you that there are 3 superfamilies of ligand-gated ion channels, which are synaptic receptors. In this mini lecture, and then the next one, we will discuss primarily the pentameric cis-loop receptors. These are the nicotinic acetylcholine receptors, the GABA-A, and the glycine receptors. Here is a single subunit of one of these receptors. It has 4 transmembrane domains. Later on, we will discuss postsynaptic glutamate receptors. Unfortunately, we won't have time to discuss the ATP receptors, also called P2X receptors. Now, let's talk in detail about the timing and the analysis of postsynaptic activation. Here we imagine that we are doing a single channel recording from a postsynaptic membrane. Single channel recordings are done with patch clamps, and here at the baseline, the channel is closed. We have one channel in the patch, and we have some agonists, some acetylcholine or some glutamate, depending on which channel we're monitoring. And every once in a while, the channel opens, it stays open for a while, and then closes. Here it opens again and stays open for a while, and then closes. What we're going to do is to analyze the timing by synchronizing artificially on the opening event. This can easily be done with software, sometimes with a bit of hardware. And so we take this final event and we line it up with the beginning of the opening. And then we do the same thing for one of the other events, etc., until we have all of the channels retrospectively synchronized as they open together. And now we superimpose them so that they are nearly all synchronized. And we see that they begin, although we have made them all begin at nearly the same time, they are closing at different times. They have a stochastic closing event. The importance of this phenomenon is that although we did this recording by taking a single channel and recording it for several minutes at a time, during a synaptic event, actually, there are many channels in parallel in the membrane. The neurotransmitter hits them nearly all at once, and so we get a macroscopic recording, which seems to be the sum of these single channel openings. So at a synapse, the pulsive transmitter is approximately a delta function, for those of you familiar with that, and all postsynaptic channels open nearly synchronously, and several drugs perturb either the synchrony or the duration of these openings, and we'll discuss them primarily in a future mini lecture when we discuss the GABA receptors.